Afternoon. So we're nearly there. So my name's David Venables. I'm CEO of Laverock Therapeutics. So it's my privilege to introduce you to the company and tell you something about our novel programmable gene silencing platform that we've been creating and validating, and talk to you about some of the programs that we're developing. So Laverock Therapeutics, we've got a very unique but powerful proprietary gene silencing technology that we can use to underpin some really novel therapeutic approaches, and I'll tell you more about that through the course of my presentation. We're utilizing the platform internally as we focus on developing our own pipeline of therapeutic products, particularly focused on the treatment of solid tumors, which, as we know, is a critical unmet need and really at the frontier of where cancer therapies need to be able to target next. One of the things there's many things that's unique about our platform, and explain what those are, but one of the key features is that we're able to create programmable cell therapies, and in that sense we're highly differentiated. Because we've got a platform that is very versatile in the way that it can be used, we're also very active on the partnering side, and talking to a number of companies about how our platform technology can be incorporated into their product design to again give some very unique therapeutic capabilities, and I'll explain more about what we mean by that. So if I start off then by explaining what our platform is, but also explaining a little bit about our philosophy of what it is that we're trying to do. So for multiple iterations now of molecular biology, what we've had in our mindset is to say, well, we've got these great molecular tools that we can build into expression cassettes, into constructs. We can develop transgenes, promoters, UTRs. We can deliver payloads, and we can put these into cells. And we hope that the cell accepts this foreign DNA that we've constructed, and hope that it performs as it was designed to perform. And in some cases we see that happens, and in many cases we don't. We actually see that the cell rejects that DNA. It doesn't like to be edited, doesn't like to have foreign DNA inserted into it. But we're trying to basically gate crash the cell's genome to our own therapeutic intent. What we're trying to do is work much more actually with the innate control systems of the cells. We know that the cells are exquisite in their ability to control and orchestrate their own gene function. So rather than trying to recreate that artificially and gate crash the cell with a system, what if you could basically work alongside those systems already within the cell and repurpose them, recondition them, recode them to have an effect that you want to have? And that's essentially what we do. That's the philosophy that we work with. So recognizing that everyone knows about microRNAs now, particularly after the Nobel Prize that was awarded. So we know that there's a level of gene orchestration that's done through families and thousands of microRNAs that are expressed or produced by a cell in response to cell differentiation, in response to environmental triggers, in response to a whole series of senses that the cell has of its environment. And that essentially becomes our raw material. So we bioinformatically, we analyze cells in certain environments. We identify these families of microRNAs. We understand the orchestration of those different microRNAs. And out of all of those thousands of potential microRNA candidates, we select the optimal microRNA that we can recode and change its targeting sequence and convert it now, rather than the natural microRNA, into an siRNA that's targeting a gene that we want to have targeted and silenced. And we can do that because there's a huge level of redundancy within these families of microRNAs. So by removing one means that the biological function of all of the cell and all of the others still is protected and still exists. And so rather than trying to take an siRNA or shRNA transgene type approach, then we go in and we basically do a minimal recode of the trigger sequence of an endogenous microRNA and redirect it to a new target. And that has huge advantages and capabilities, which I'll summarize in the next slide. So this means that we can orchestrate and engineer cells to orchestrate programmable, tunable, stable, specific, and we've now also shown multiplexing gene silencing under very specific and selective conditions. And this sets us apart from classic genome editing or other siRNA oligo-based gene silencing approaches. And this underpins the power, the novelty, and the versatility of our platform. So the question then is, well, how do we apply this, and where do we see utility for this, and what are we actually focused on? So as a company, there's many things that we could do. So what we've chosen to do is focus on a few key targets. The first is ex vivo in tackling solid tumor and the tumor microenvironment, the suppressive tumor microenvironment. So we can edit and engineer CAR T cells, so we can engineer specific properties into T cells to overcome many of the restrictions on on-target effect, efficacy, and persistence. We're also developing a macrophage program that we're targeting to solid tumors, and I'll explain more about these shortly. We've just heard an intro to some of the approaches in type 1 diabetes. What we've done is take an approach where we can actually create programmable silencing of immune HLA factors, so we can actually create a hypoimmune cell line where the hypoimmunity is actually linked to the cell identity. So there's no concerns over de-differentiation of cells or mixed populations of other non-target cells within your product when you implant it. And we also see huge potential for the platform itself in an in vivo editing approach, particularly in diseases like CMT1A that are the result of overexpression or gene duplication leading to overexpression, where tunable silencing of that gene is actually critical to bring it back into the sweet spot of gene expression. So you can't do a knockout. It's not needing to replace or supplement a gene, but you need tunable silencing within specific cells. And we can select microRNAs specific to those cells that we can recode to give a predictable level of gene silencing. And that's our CMT program that we're working on, which I won't talk too much about in this presentation. So if we think then about the CAR Ts, which everyone's obviously very familiar with, and our question is, well, how can we use our platform to enhance persistence and also TME resistance? So there's a number of different ways that we can do this, and maybe I'll just, in the interest of time, just give you maybe one example, and that is CAR T cells that have had checkpoint pathways knocked out or inhibited to increase their tumor killing is a standard approach that's taken. With our technology, what we're able to do is identify microRNAs or families of microRNAs that are expressed in CAR T cells only when that T cell, that CAR, engages with its target. So as the cell engages with the target, we see a profile of microRNAs that's upregulated, and when that CAR disengages from its target or the tumor is cleared, that family or those profiles of microRNAs drop back down to non-expression or basal levels. So you've got a built-in programmable microRNA within CAR T cells and within activated CAR T cells. What we've been able to show is that we can edit those microRNAs in a multiplex way so that we can get multiple checkpoint pathways inhibited within that CAR T cell only when that CAR T is engaging with its target. So it's only under that circumstance that the checkpoint pathways are downregulated or silenced. And this increases, obviously, the safety of those products. It also addresses and increases persistence as well. And so this is one of our first generation products that we're working with. And then we have a similar or an approach that also where we're repurposing microRNAs to target specific classes of HLA to create an allogeneic cell therapy or allogeneic CAR T. So the concept, so this is an area that we obviously provide with our platform a series of benefits. So the simultaneous silencing of multiple inhibitory pathways, and all of this involves minimal genome editing. And so we don't end up with an over-engineered and exhausted T cell. It's very subtle and minimal editing that has to be undertaken to achieve this. And so we maintain cell fitness, we maintain cell potency, reduce manufacturing complexity, but we get this programmable silencing feature. And so this is an area we're actively talking to companies about partnering in. We're not developing cars ourselves, so we're keen to talk to anybody who's working with cars and TCR design and has validated targets who would be interested in partnering with us on this specific program. With our macrophage program, then, this here, rather than starting with donor-derived cells, what we're starting with is iPSC cells. And what we can do is we can go into an iPSC cell and recode microRNAs in that iPSC cell that would only be expressed when that cell is differentiated into a macrophage. And we're able to do edits of microRNAs that don't perturb that differentiation protocol. And this again shows how subtle our approach is in terms of its impact on the biology and the functionality of the cell. But when that macrophage or when the iPSC cell is differentiated into a macrophage, it's pre-programmed to actually drive that macrophage into a pro-inflammatory state. And so we have identified particular gene targets within the macrophage that enable us to do that, even to the extent that if we expose that macrophage to TAM-like conditions that should drive it to a TAM state, it still actually resists that polarization and will only polarize towards the pro-inflammatory state. And so this now is part of a platform that we're developing to develop a suite of macrophages and macrophage concepts that we're taking through. Currently, we're in vivo studies in tumor models, and we have more data coming out to support these concepts over the next 6 months. So watch this space. So we can see macrophages as a tool to be used to tackle solid tumors. We can supplement those macrophages with other features as well as the polarization state, and we can use our technology to create hypoimmunogenic macrophages for an allogeneic therapy as well. Interest of time, I'll skip over this, but again, delivering on the tunable, multiplexed, and programmable gene silencing gives us a competitive advantage in the macrophage space. The other area that we've also been working on, we've talked about hypoimmunogenicity. The key feature, again, here is what we're doing is we're identifying microRNAs that are specific to cell identity. So if you're differentiating cells from iPSCs to a target cell, like a islet cell for a type 1 diabetes program, what we're have been able to do is identify microRNAs that is specific to that differentiated cell, and then reprogram those microRNAs to have very specific HLA silencing effect. We've done it both with tunable silencing of beta-2 microglobulin, but we've also done it with very specific silencing of HLA subtypes that enable us to get specific, for example, HLA-A silencing while leaving HLA-E intact, which is obviously required for the immune system. So, but those cells, if they then differentiate away from or de-differentiate from that state that you want them to be hypoimmunogenic in, that hypoimmunogenicity is lifted and that's lost. So it adds an extra safety measure to cell therapies and hopefully gets us away from the need from a lot of the encapsulation type technologies that we're currently subjected to. So that's a very brief overview of the platform, the technology. We've got a strong team that we've built together. We're at a seed stage company. We raised our seed fund a couple of years ago. We've just been successful in extending that seed fund just in the last few weeks. So more news on that coming up shortly. We've built and industrialized the platform so we can do the analysis and the engineering at scale. So we're set up for high throughput, which again, I've not talked about that in any detail, but we've got a significant and high throughput platform to be able to develop and validate these targets and develop these cell lines. We've got a clear strategy to develop our own therapeutics and take those forward, but also to partner the platform with others, because as I said, it's hugely versatile and can be used for many things. And we've been successful in our fundraising to date as well, which is very exciting. So on that point, thanks very much.